The first slide of this lab is a section of rabbit liver that has come from a rabbit infected with Eimeria stiedae. And this is a rather confusing slide in a number of ways because of the level of infection that is present in this slide. One of the easiest ways to first get oriented in this slide is to look at some of the normal liver tissue that is present in each of the slides. And typically you can find in this areas where there are just uninfected liver hepatocytes with the typical cytoarchitecture of the liver, with central veins and with bile ducts present. This particular infection parasitizes the cells of the bile duct, and it originates in the large bile duct, and it will eventually extend further up into the bile duct, the smaller bile ducts that run through the liver. This would be, at the end of the pointer, the typical appearance of that uninfected bile duct. However, by the time that the infection has progressed only slightly, the bile duct will start to undergo hypertrophy, and you will see an enlarging of the bile duct as indicated at the end of the pointer. However, with time, those cells will continue to undergo hyperplasia and hypertrophy to the extent where the whole field of view can be occupied by what was previously just this small bile duct. And everything in your field of view right now is what originated from a small bile duct. However, the number of cells have increased to a very large degree, and this is presumably in response to the parasite's need for additional cells to infect. They're tricking the host cells to do so. And so at this magnification, you can see how extensive that bile duct has become. Inside in the lumen of this bile duct, you will find large numbers of oocysts. These oocysts will pass down the bile duct and into the intestine and be excreted out from that. If we go to a higher magnification, we can start to see a little bit more detail in terms of the appearance of these oocysts. They have a rather thick wall, and you can see usually a dot in the center of many of them, which is from the nucleus of the ova. We can also start to see the cells of the bile duct. These are, at the end of the pointer, uninfected cells, and then in and amongst those uninfected cells are cells infected with primarily macro and microgametocytes. Using the 4DX objective, you can begin to see even more detail in and amongst the various cells. And again, you can see at the end of the pointer, the uninfected epithelial cells of the bile duct. And then you can also see various levels of development for the macrogametocyte. At the end of the pointer would be a fairly immature macrogametocyte. They will then begin forming these globules that lie along the outside, nucleus in the center, in these more mature but probably unfertilized macrogametocytes here. After the macrogametocyte becomes fertilized, it will start to lay down that oocyst wall through through those globules that are surrounding the outside, and eventually then dumping out into the lumen of the bile duct and traveling on down. Now it's a little bit more difficult to find the macrogametocytes, the microgametocytes, and it's a little bit difficult sometimes to differentiate those. Typically what you will see is a cell that's filled with very large numbers of black dots, each of the black dots representing a microgamete, and at the end of the pointer now is one of those microgametocytes. Because this is a fairly old infection, most of the infected cells are containing just the micro and macrogametocytes, and it is extremely difficult to find schizonts containing the merozoites. At one point early in the infection, most of what you would have seen were the schizont stages, and so I'm not going to ask that you find any of those.